Hello and welcome to BioRules video lessons. Today we are going to be talking about biotechnology or gene technology, the technology of playing or using or identifying or something related to genes. This gene technology, which is part of the biotechnology, is going to be involving some parts of this biotechnology here in our course. The first one is going to be DNA profile, which is basically doing a profile, doing an identification or trying to do a matching of DNA with a sample. So basically this is used with crime scene investigation and paternity tests. This is basically the class today. Genetic screening also uses DNA profiling, but instead of comparing to a sample like a crime scene sample or a potential father or mother, it checks for markers of disease. So to see if someone has a genetic disease or some genetic disease, you screen that person to see if it matches. Also use the same technique that we're going to be using today. Okay, so basically it's looking for a disease or condition that is genetic. Gene transfer, basically this here is the genetic modifier organism, transgenics, so you're going to see that in the future. Cloning is also part of biotechnology. The Genome Project is part of biotechnology, and there are other things that we can study that are part of gene technology. Today we are going to be taking a look on what to do for a DNA profile or a DNA screening. So basically what to do when you want to look for a gene. So when you want to look for a gene, the first thing that you have to do is to get a sample. So you get material that has cells, hair, saliva, blood, semen, skin, whatever material that's going to be having cells, and those cells are going to be having DNA. So for paternity tests, we can get a fair amount of material, saliva, blood, whatever, but in crime scene investigation, the volume is going to be a problem. It's going to be very, 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 very small. So usually, we are going to be needing to be magnifying the amount of DNA that's around. So basically, we are going to be needing to do replication. So DNA replication, if you remember from when we study genetics, when we study actually DNA, replication is done by the cells and basically consists of opening the DNA with an enzyme called helicase, and then adding a primer with RNA primase, and then DNA polymerase is used to add new nucleotides, and depending if it's 5 to 3 or 3 to 5, it finishes. But basically those are the steps. When we want to do a magnification of material, but in a lab, we are not going to be counting on living things because they take too long. It's quite complicated. And there is an easier way to be doing this replication. And this is using a technique called polymerase chain reaction, also known as PCR. You are very aware of PCR. You have done probably PCR during these pandemics to identify viruses. And PCR actually is a technique that instead of using living things, they are going to be using a machine, a very simple machine, very elegant functioning machine that will have a series of steps. This machine, by the way, is called a thermocycler because it's going to be cycling the temperature where this DNA is going to be located. The first thing is to separate the strands. Without using helicases, which is biological, and then you cannot control when helicase is going to be working or not, we use heat. If you warm up to 98 degrees Celsius a fragment of DNA, the covalent bonds are not broken, so these guys here, they stay intact, but the hydrogen bonds in between the two tapes, they are denaturated, so basically they are going to be separating. So the first step in a PCR is to warm up the genetic material. After that, we have a step called annealing, which can be used to already manage to make a PCR with fragments of DNA on the size that we want. So the PCR can not only magnify, but depending on the primers you use, can also already separate in fragments of size or different size or something like that. But we need to have a primer. Instead of putting a primase that will make an RNA primer, we already put a certain primer that you start copying, and that's going to be DNA. So it is part of what will go into this machine, a certain DNA primer. And that, of course, is going to be colder, because otherwise the new primer is not going to be sticking. Usually this happens at a temperature of around 52 degrees Celsius. After that, DNA polymerase has to work, but it's not any DNA polymerase this polymerase is not your polymerase because the solution where this stuff is going to be, it goes to 98 degrees Celsius. Your polymerase, which is a human enzyme, would have been denaturated. So use a polymerase called Taq polymerase. It's a polymerase that have been extracted from hot springs bacteria. So they are able to stay stable even at very high, almost boiling temperatures of 90 degrees Celsius. After the annealing that happened at around 52 degrees Celsius, we warm up a little bit more to around 72. It's the optimum temperature for the Taq polymerase to work, and the polymerase does its job, just copy the DNA. And voila, we have one copy of DNA just by warming up to 98, reducing to 52, and going to 72 degrees Celsius. So basically, this here is the cycle that you're going to be having. Warm up to 98, cool down to around 52 to have the annealing, and then go to 72 where we have the extension part of the process, all that inside of a thermocycler, which is the PCR machine. And the elegant thing here is the following. If you do that in the first cycle, where you had one copy of DNA, now you're going to have two copies. Actually, if in the zero moment you had two copies, now you have four. But if in the first one you have one copy, you're going to have 2, 4, 8, 16, 32. And the beauty is the following. After about 30 cycles, we are going to have around 1 billion copies of DNA. 1 billion. That's a very big amount of DNA. It's not a very big volume of DNA because DNA, although it's very long, it's very thin and very compact, but it's going to be 1 billion copies of DNA. So that's quite a lot of DNA that have been magnified with only around 30 cycles. And that takes around 1.5 hours to 2 hours in this process. So it's a reasonably fast process to make a huge amount of DNA. Okay? The idea, just to illustrate, is going to be something like that. You have a piece of DNA. After the first cycle, we have 2 pieces of DNA. And then in the second cycle, we are going to, each one of those is going to make 2 more. So where we had 2, now we have 4 pieces of DNA. And where we had 4, in the third cycle, we are going to be copying and we're going to be ending up 8 pieces of DNA. So it's going to be 2 to the power of 1, 2 to the power of 2, 2 to the power of 3. And after 30 cycles, we're going to have 2 to the power of 30 pieces of DNA. Okay, so I'm not going to be waiting for this to finish copying, but 2 to the power of 4, after 30 cycles, 2 to the power of 30 DNAs. This is PCR, is the second step after collecting the material for doing the DNA profile or identifying piece of DNA. Another step that may be just the use of primers in the PCR, just use of primers may already separate DNA into different pieces and sometimes pieces of different sizes in different people. We can also use restriction enzymes. Okay, I'm going to illustrate to you what happens here with restriction enzymes, but bear in mind that this here could also happen during the adding of the primer itself in the PCR. Okay, so what's going to be happening here is the following. There is this restriction enzyme, which is an enzyme that cuts DNA in a certain sequence. In this example here, it cuts where it is an AAA TTT and the complementary TTT, TTAA. That thing is going to be cutting here. In this example, this enzyme actually makes a weird cut. Let me do this in blue here. It's going to make a weird cut that cuts actually over here, over there, and here. It does a weird cut over there. We'll see more on this weird cut that not necessarily is going to be a blunt cut over there when you talk about another biotechnology, which is the transgenic technology. Okay, there are different restriction enzymes. For example, here you have one that cuts in the AGCT. This here is GGCC. This here is GGATCC, and so on. There are many restriction enzymes. They cut in a certain sequence. And the beauty here is the following. You are going to have in your DNA places where you have a certain sequence. For example, in this example, we have an AATT over here and an AATT over there. So adding this restriction enzyme, we just cut the DNA in 2 pieces over there. So we cut the DNA into a fragment of a certain size. And what's interesting is that maybe different people may have this different sequence AATT in different places of DNA, cutting DNA in a different size. So for example, here we have a restriction the nuclease that cuts DNA in the same place in everybody. This is very useful when you want to separate a certain piece of DNA of the same size to be worked with, within everybody. That's going to be quite useful in biotechnology of transgenics. And it can also cut in different places with different people. So here, for example, you have AATT, for example, in different position in different people. So it's going to be making fragments of different sites. So because people have letters sometimes in different places. The idea here is that this restriction nuclease is going to be cutting these fragments that are going to be workable. So it's going to be cutting a certain piece of a certain size that can be workable, and you throw away the rest. And then we use the non-specific, the one that can cut in different places, in different people, in different piece of DNA on different size. And then you're going to end up having DNA of different size. And the beauty here is the following. If you want to compare a DNA of a person with a sample, you just have to see if the fragment size that you cut using the same restriction enzyme, if the size of these fragments, if they match. If they do match, then you have the same sample of DNA in both of them, in the person, the suspect, for example, in the crime scene, and so on. So this here is how it's going to be working. Okay. After we cut the piece of DNA, or when you do a PCR already with primers that make fragments of DNA of different size, what you're going to be doing next is going to be using a technique called gel electrophoresis, where we apply an electric field to make DNA to run from one side to the other. So basically, we put a negative pole, we put a positive pole, and the DNA runs toward the positive pole. So it runs in the gel from the place where you have put it in the beginning, that was on the top over here. The idea is that different sizes of DNA, they don't manage to run with different speeds or with the same facility, the same speed on the same gel. The gel makes it difficult for piece of DNA to run. So as a result, a small piece of DNA usually run more. So ending up that they end in this here. So the bottom line here represents small pieces of DNA. And large pieces of DNA don't manage to run so much. So they are going to end up over here. So we are going to be ending up having bands of DNA. And these bands of DNA represent size of DNA. The ones that run more toward the positive side is going to be smaller, and the one that runs less is going to be larger. This is the idea of gel electrophoresis. Then we put ultraviolet radiation to be able to see the DNA, and we can take a look on the pattern of bands. So here is this here is showing things illustrated. We have here 3 samples that were put in the slots close to the negative pole. We turn on the positive pole over there, and we turn on the electricity, and the DNA is attracted to the positive side and is separated with the small pieces moving a lot and the large pieces moving less. And then by the pattern of these bands, we are capable of identifying DNA fragments. Just to give you an idea here, we have 4 different restriction enzymes, the restriction enzyme G, the A, the T, and the C, that have been run on crime scene and also have been run on suspect number 1 and suspect number 2. And what this here shows us is that neither of them, the G, the A, the T, the C, they do not match with suspect number 2. So suspect number 2 is not associated with this material that was found in the crime scene. Suspect number 1, on the other hand, it matches for the G, for the A, for the T, for the C. So 4 times matching. This is not a coincidence. And then we can say, okay, suspect number 1 is associated with the crime scene. We cannot say that he committed the crime, but we can say that his DNA was found in the crime scene. Okay. Another example here, we have material in the crime scene and suspects number 1, 2, and 3. In this case, many restriction enzymes have been used to make many different fragments in there, and we end up having suspect number 1 not matching, suspect number 3 not matching, but suspect number 1 matching perfectly the material that was found in the crime scene. So suspect number 2 is associated to the crime scene. Okay. Another example here, blood stain from a certain place. It could be a crime scene, and we have here for there we have with a line over here, we can see that the material matches, all of the material on the bloodstain matches with John. It doesn't match everything in Bob, so there are some that's actually one missing in Bob here and one that's not present in there in the bloodstain. So it may mean that Bob is a relative of the suspect, but not the suspect over there. Sue doesn't match, so Bob doesn't match, Sue doesn't match, John matches, and Lisa doesn't match. So in this case, the blood stain belongs to John. Okay. We can also do paternity tests, but in case of paternity tests, the child's DNA is not going to be matching completely with the mother or the father, but it must match with both of them, mother and father, in half of the places. So basically what we're going to be having here is that this fragment of DNA in the child matches with the one in the mother. This fragment of DNA here in the child matches the one with the father. This here matches with the mother, this here matches with the father, and this here matches with both of them, which may mean that the child has it from the mother or from the father, or actually the child can have two identical copies in there, and then we can say that yes, this is the mother and this is possibly the father because they match. This is a couple that could be the parents of the child. Okay, another example here, we have a child, we have Mary, which is the mother, and we can say that this here matches with Mary, and then we have this here not matching with Bob, but matching with Larry. So this here matches with Larry, this one matches with Larry and Bob, and this one here matches with Larry. So the only parent, Bob or Larry, that matches with the child, and the ones that Mary doesn't match is Larry. So Larry here is the potential father. Hey Larry, you are the dad. Okay, I hope you have enjoyed this. Let's talk more about that in class, and you have a quiz now to do, and see you in class. Bye bye.